I'm Kate, and along with Katie and Ariana, we're going to be talking about genetic sequencing today. So through this project, we really hope that you guys will be able to not only understand genetic sequencing, but also understand the impact and influence that it has on medicine and healthcare today. So just to go through a little bit about genetic sequencing, with the 4 different bases and nucleotides, there ends up being a really large scale because there are 3 billion in the genome. So with that, there's 3 different types of sequencing. This goes through the Sanger sequencing here. And Sanger, along with the Illumina sequencing, really increased the efficiency because it was a way to amplify PCR to get enough data. So this here talks about the Sanger sequencing. So now just to go through a little bit of the timeline with this Sanger sequencing, what's the big deal? So we want to make a way that's cheap but efficient. So we want a way to lower the cost but also have faster readings and still maintain that accuracy. So here talks about a little bit about that Sanger sequencing. So in that, we're really talking about a way to still use single sequencing DNA methods, but we're using ddNTPs to stop the elongation. So through that, you end up having to use gel electrophoresis to separate, and then you use bands to determine the different time lengths of the DNA sequencing. So this is called the next generation sequencing. This is one of the first methods that was used for genetic sequencing, but we're on now to next generation. So now we're talking about expanding the applications of genetic sequencing. So it's now being used for IVF and also being able to test for microbes that are attacking humans. So because of genetic sequencing, we're now able to go back and sort of help prevent misdiagnosis with genetic sequencing. So just to talk a little bit about the different machines that were developed. So the 2 different things with the Sanger sequencing and then the Illumina were 2 different machines that were really landmark in developing a way to genetic sequence that was cost efficient but also was able to be efficient in a certain timeline. So basically it goes through the different lengths here. So now on to nanopore technology. This is the 3rd generation. This is the newest technology with genetic sequencing. So you're able to detect the DNA when it goes through a smaller size pore by an interruption in the voltage. So basically, we're still evaluating this with new biological engineering, but at the same time, we still need to scale this on. So to talk a little bit more about nanopore technology, there are some pros and cons with it. It is the latest technology, and we're able to really use it because you don't need to amplify using PCR. So it's really a lot more efficient and also cost efficient, but at the same time, we don't have enough of engineering to really figure out how to make it more cost efficient. So now we're taking a step back and going back to this 2nd generation, which is called the next generation Illumina sequencing. So the problem with this is that you have to add fluorescent nucleotides to it, which is really cost inefficient. But at the same time, you are able to get a huge number of reads in genetic sequencing because it goes with a huge number of nucleotides with 1 short read and 1 stroke. So to talk a little bit more about Illumina sequencing, they use 100 base pairs, and then those base pairs are amplified using PCR. They're then separated into single-stranded DNA, and then DNA polymerase and nucleotides that are fluorescent are added to that to label it, and then you take an image of the slide. The problem with this is that you have to go back each time to take a new image. So then to talk a little bit, continuing about Illumina sequencing, what ends up happening is with each terminator that's removed, then the fluorescent label is removed as well. So it's sort of a one-at-a-time sequence that's going on. So in that way, we're still using this for smaller scale projects, but because it's not efficient as this next generation nanopore technology, it's not used as frequently. But there's still a lot of implications of this. So it's a way, this new genetic sequencing is a really great way to develop personalized medicine, not only in pharmaceutical industry, but also in personal healthcare coaching, because it's a way to take people that have predispositions to diseases because of their genetics and then really evaluate that and go back and develop a personalized approach to that. So to talk about some of the ethical implications of that, there really are a lot that involves privacy of all of your genetic sequencing. Are we going to allow the public to have access to all of that? The cost. We're now down to close to $1,000 genome. Before it was in the billions, then it went down to 10,000, but not everyone has the availability and accessibility to it. So we are still figuring out everything with the ethical implications. Not everyone has accessibility to it in terms of the cost of it. The genetic sequencing has been outsourced to China now, so that does help lower the cost a little bit. But it does have a big influence on business, biotechnology, personalized medicine, customizing drugs and treatment for that as well. So all of these different genetic sequencing, these basically 3 different techniques that we're now onto this nanopore technology have really affected a lot of different industries in pharmaceuticals, healthcare, business, biotechnology, and everything as a way to minimize long-care risks of disease and predisposition to disease. So now we're going to go into some of our propositions and discussion. So my other group members are going to come up. Sorry, that was super fast. But we really want to, some of the main goals that we hoped to accomplish with this presentation was to really talk about how the different implications of all of these.
Okay, I'm going to stop you.
Okay.
Okay? So
Okay.
it's up for the next person now to introduce the proposition.
Okay, sounds good.
Thank you.